L.L.S.Seniors 2, and now we are going to explain a new lesson. Okay, our today lesson about enzymes. Okay, enzymes first, it's a catalyst. So enzyme is a catalyst. What is the difference between enzyme and catalyst? Okay, enzyme its structure is a biochemical structure, so it's organic structure. Okay, biochemical meaning organic structure. The structure of enzymes is a protein. While catalyst, okay, its structure is a mineral structure. Catalyst is a general, okay, name about any substance I can add it in the reaction and accelerate the chemical reaction. So the difference between catalyst and enzyme, okay, both of them accelerate the reaction. Meaning of the acceleration of reaction, okay, number one, reduction of time. Okay, number one, reduction of time. Number two, what also fast or make it rapid the reactions. Okay, so we have here a lot of difference between enzyme and catalyst. Catalyst is general, meaning, okay, while enzyme is a part or type of the catalyst, okay, but all of them, both of them accelerate the reaction, meaning of acceleration, reduction of the time, and make it fast. Without the enzyme, the reaction will be occurred, but will take a lot of time, spend a lot of time. So we have here and very important for enzymes. Okay, enzymes are proteins. Enzymes are proteins. So has the same structure of the protein. Okay, can you remember the structure of protein? The structure of protein was amino acid, and the amino acid is carbon with the 4 arms, hydrogen here, carboxyl here, amine group here, and last one are alkyl group. So enzyme is a protein, okay, not verse. Enzyme is protein, not all proteins enzymes, okay, not all proteins enzymes. So the first is fact, the second one false. All enzymes are proteins. All enzymes are proteins. Okay. To start any bioreaction, you need energy. Any reaction, if it catabolic reaction or anabolic reaction, you need energy. Okay, so this energy may be high energy. So we have here another concept is activation energy. Meaning of activation energy, it's energy that the reaction need to start the reaction. Energy to start reaction. Okay, this energy, a huge amount of energy. Okay, can you imagine your body spent a lot of energy that can be needed in a lot of different parts inside your body in only one reaction? Okay, how can I reduce this reaction? Reduce this energy by adding of enzyme. So enzyme here will reduce what? Activation energy. Enzyme will reduce activation energy. Enzyme will reduce what? The time. Enzyme will accelerate the speed of the reaction. So those the missions of enzymes. Okay, give me a graph, and graphs here very important, okay, in our lesson. Enzymes, as we said, reduce the amount of energy that the reaction need to be half or more. Okay, look at this graph. Activation energy. We have here two graphs. First one with high rate and second one with the low rate. Which one with enzyme? Which one absence with enzyme? Look carefully. This activation energy needed more. So enzyme here not share. Minus of enzyme here absence. By adding enzyme, the amount of activation energy be decreased. So here with the enzyme present over enzyme. So we have here two curves on the same graph. One of them point to the presence of enzyme, while the another one points to the absence of enzymes. Okay. So enzyme make three rules, as we said: reduce time, reduce activation energy, and accelerate or increase the accelerate the speed of reactions. Okay, now I want you to know the main reactions that occurred inside your body. The one reactions that occurred inside your body, we can summarize them into bioreactions. Meaning of bioreactions, a total reaction that's occurred inside your body. Bioreactions is named by metabolic reactions. Metabolic reactions is divided into two parts. Okay, first one, catabolic reactions, and second one is anabolic reactions. Meaning of catabolic reactions change from polymer to monomer. So catabolic reaction change from polymer to monomer. Inside your body, from polymer to monomer, okay, we have to process represent that. First one, digestion. Yes, digestion. By digestion, I can change a lot of types of food like carbohydrates, proteins, and lipids into what? Carbohydrates. Okay, carbohydrates can change to monosaccharides. Proteins, amino acids, lipids into fatty acids plus glycerol. So we have here digestion. Digestion refers to process to catabolism, then polymers inside your food, inside your diet, into monomers, monosaccharide, amino acids, and fatty acids with glycerol. Okay, what can I do by those? Okay, they will not stay inside your digestive system. Okay, they will turn to another machine that can use them for first one, monosaccharide. Monosaccharide get inside cell and make the second step. Second step is oxidation. Yes, oxidation will burn, oxidize the monosaccharide to release energy, release energy. And fatty acids also get inside the cell to release energy. So we have here second step for catabolism, okay? Number one, digestion. Number two, oxidation. Oxidation meaning get the monomers inside the cell to release energy. We have here another sequence of reactions that occur but in opposite directions. Anabolic reaction. Meaning of anabolic, okay, I can get those monomers and turn them to polymers. Yes. To form from polymer to monomer, you release energy. Please don't forget. From monomer to polymer. Monomer like amino acids. Like monosaccharides. Amino acid can change into protein. Can change into protein. Monosaccharides can change into glycogen. And the last one, fatty acids. Fatty acids with glycerol plus glycerol can change into fats again, or back to fats and store all the information under skin and share in cell building. Share in cell building. Protein help in building bodies, building tissues, building bodies and tissues and muscles and so on. Glycogen, as we said before, stored in liver, stored in muscles. So we have here two types of reactions. First one, catabolic. Second one, anabolic reactions. Now, I want you to know the main idea of enzyme. Enzyme share in these reactions. Metabolic reactions that occur inside your body cannot perform in a high quality one without the absence of enzymes. So we had here, we need here enzyme to complete our mission, metabolic process. So metabolic process is total reactions of catabolic and anabolic. Give me an example on catabolic and anabolic reactions. Catabolic reactions meaning of turn from polymer to monomer. From polymer to monomer, number one, as we said, digestion. Number two, cellular respiration. Meaning of cellular respiration, glucose monosaccharide get inside the cell to release ATP. What is ATP? It is that energy. So, cellular respiration, both of them and digestion represent as, catabolic reactions. Anabolic reaction, the other side, meaning change from monomer to polymer. Monomer to polymer, okay, like building of your tissues. One, building bodies, okay, repair damages tissues, okay, now photosynthesis process. Number three, formation of glycogen. Oh yes, all of them are anabolic process. Okay. Now we are going to explain more about enzymes. Enzyme, as we said before, is a protein. Okay, this is first information with you. And also, enzyme consists of protein. Let me remember you. Protein So consists of two main parts. Acidic part and basic part. Acidic part CHRCOOH is carboxyl acidic part, while another one NH2 is the basic part. So the nature of enzyme is neutral. Why? Because of the presence of two sides of acidic and basic parts. Now we are going to know the main properties of enzymes. Enzymes, first of all, increase or accelerate the bioreactions. Number two is specific in its work. Meaning of its specific properties of the enzymes. Number one, acceleration is with you. Number two, what? Enzyme specific in work. Meaning of specific, any enzyme consists of active site. This active site is the where or the place where the S, meaning of S, substrate or substance, So S is the substrate or substance. Enzyme deal with the substrate, so the reaction will be occurred. If enzyme cannot occur or cannot work to or with the substrate, the reaction cannot be occurred. Give me an example. Okay, a enzyme is specific S. So enzyme want one S can get inside this active site. After this, it will give ES. Meaning of ES is intermediate structure. But this intermediate structure is unconstant, unstable, unconstant or unstable. Later, this complex will turn to first one. Enzyme will get out without any change in its properties. Same volume, same inner properties. While the substrate S will change to products. These products will be S1 and S2. Meaning of that, substrate, the complete substrate, will be broken into two products. So the enzyme here gets out without changing any properties or its volume. Okay. So the main properties: number one, acceleration; number two, specific in work. To perform its work accurately, have to the active site same the substrate. So it's like lock and key theory. Lock and key, if the key same in lock, so the lock and the key will be tied together. If it not, cannot open the lock. Okay. Now we are going to know the meaning of limiting factors. The means limiting factors that affect the activity of enzymes. Limiting factors. We have 5 limiting factors. First one, concentration of the enzyme. Number 2, concentration of the substrate. Number 3, pH or power of the hydrogen. Number 4, temperature. And the last one, the presence of inhibitors. Those are 5 factors that detect any activity activity of enzymes. Okay, meaning of inhibitors. Inhibitors meaning something add to the solution that resists the work of enzyme. Enzyme accelerate the reaction, while inhibitor resists this acceleration. So inhibitor resists the work of enzyme. Okay. Concentration of enzyme, concentration of the substrate. Concentration of enzyme and substrate, the concentration of enzyme and substrate have to be equal. If I want 1 milligram of enzyme and 1 milligram of substrate, 1 milligram of enzyme will activate 1 milligram of substrate, and the reaction will be okay. If I have 2 milligram of enzymes and only 1 milligram of substrate, so not all enzymes volume will work. Only one will work on 1 milligram of substrate. So the amount here is very important. Okay. If they are equal, the reaction will, it's acceleration of reaction. Okay, first the acceleration of reaction will increase till one of those and the reaction will stop. Okay, this is diagram that represents the concentration of enzymes with the substrate. Now we are going to explain the now concentration of enzymes, substrate, and inhibitors. Now we are going to explain temperature. Temperature is a very important factor that depends on the work of the enzyme. Any enzyme to work has 3 temperatures. Look here carefully. 3 temperature. By this diagram show the relation between temperature and activation. Okay. First temperature here at which the reaction starts, named by minimal temperature, the least temperature at which the reaction starts up. And we have here what? Optimal temperature. It is the highest temperature, highest quality of the reaction. Okay? Optimal temperature is the perfect temperature at which the enzyme can work by all effort. And third one here is the maximal temperature, which is the highest temperature, highest work temperature. So we have here 3 temperatures. The distance between the minimal and the maximal, okay, named by range of the temperature. If it were, okay, 0 and 35, that's 7 and 40. So minimum here 0 and optimal 37 and the maximum is 40. Okay, the temperature by increasing first from minimal to optimal, the reaction increase speed till reach to the optimal temperature. After reaching to the optimal temperature, it will decrease again. So the activity of enzyme not increase all the time by increased temperature. Okay, till reach only optimal temperature. After this will be decreased till reaching to the maximum temperature. After reaching the maximum temperature and the body is still increasing temperature, okay, after the maximum temperature, the enzyme cannot back to its activity by decreasing temperature. So by decreasing temperature again, the enzyme cannot back to its activity. Why? Because of denaturation. Meaning of enzyme denaturated. enzyme by stealing, increasing temperature, okay, we have here process denaturation. Meaning of denaturation, enzyme exchange its structure. If the enzyme changes structure, it cannot do its work perfect, so cannot accelerate the reaction and the reaction will stop. Okay. Second fifth factor, pH. pH or power of hydrogen, also very important factor. We said before, enzyme is neutral because of the presence of carboxyl acidic one and the basic amine one. Okay, so we have here two sides, but also we have here graph for pH, okay, and this pH and activity. Curve also like curve of temperature, okay, please focus. Start from minimal, optimal, and end with the maximal. First, I want you to know the scale of pH meter. pH meter. pH meter starts from 1 till 14 by intermediate point 7. Before 7 is acid, after 7 is alkaline, and each enzyme needs the amount of hydrogen to know the perfect medium to work, if it's basic or if it's acid. Okay, if you start from 1 till say 7, and the perfect one is 5. Okay, so by increasing the pH, the activity will increase to reach the optimal one, 5. After this, the reaction will decrease to reach to 7. By increasing the pH after 7, if we reach to 8, and I want you to decrease the pH again, it will be like temperature cannot back or will back. It will back. So by decreasing pH, the enzyme can reactive again back to its activity. It's very important. Okay. Give me an example inside your body enzymes that work in basic or acidic medium. Like, first, we have pepsin enzyme, trypsin enzyme. Pepsin enzyme starts inside your stomach, so the location and pH. Location of pepsin inside your stomach, trypsin inside the small intestine. The pH of pepsin from 1.5 to 2.5, so it's acidic medium. Trypsin or trypsin from small intestine from 7.5 to 8, so it's alkaline. Okay. Before 8 alkaline. So we have here two enzymes inside your body. Both of them work on protein, digest protein, and turn into polypeptide. So after the protein with pepsin and trypsin enzymes, turn into polypeptide. So the structure here, same of the results of products, but the pH is different. We have here another enzyme inside your body, amylase. Amylase inside mouth, but it works on the starch in pH equal 7.5. By dealing starch with amylase, starch change to maltose disaccharide. And we have here very important experiment. Please focus with me. We have here three test tubes. A, B, and C. Both of them I will put one starch, two amylase. And putting buffering solution. Meaning of buffering solution is solution that control pH. Buffer solution here is at 7.5 equal. Here, buffer solution more than 7.5. Buffering solution here less than 7.5. Okay, and after I will put what? Iodine solution. Number one, amylase enzyme work in 7.5. So amylase will work here. After working, starch will disappear and change to maltose. Here, buffering more and less than 7.4, starch cannot work. So starch is still here without digestion. After putting iodine searching for starch. If it found it, the color of the starch will change color to blue. Here, starch vanished, maltose, so the color will change. Now, still orange as it. So here we have a very important point about the pH. pH control the working of enzyme. If it reach the optimum pH or start point of pH, it will work. If it not, the enzyme cannot work. Okay. Thank you, Senior 1, and see you again, inshallah, in next video.